Welcome everyone to our second pitch session of the day and also our second session featuring therapeutics companies. If you missed the first session, you can watch it through the recordings on the platform. We had some really interesting companies present. This session is sponsored by Gowling WLG, and to introduce today's companies, we have Lisa Thorne, a partner with Gowling WLG, with us. Welcome, Lisa.
Thank you, Wendy, and good morning to all of our attendees and speakers. My name is Lisa Thorne, and I am a partner in the Life Sciences Group in the Vancouver office of Gowling WLG. My practice focuses on patent prosecution in the chemical and life sciences industries. Gowling WLG is very excited to have the opportunity to sponsor this session, and we look forward to hearing from the 5 companies: Bold Therapeutics, Mesintel Therapeutics, Q Biologics, Serogene Therapeutics, and Sustain Therapeutics. It is a very exciting time in the life sciences sector in British Columbia, and Gowling WLG is proud to be part of this ecosystem. We have a global team of almost 200 intellectual property and life sciences professionals, many of whom have advanced scientific degrees and have worked in the life sciences industry, enabling us to understand the opportunities and challenges that are faced by life sciences companies. We support our clients throughout their entire intellectual property life cycle, and our international growth allows us to manage global portfolios for our clients. We have expertise to support all services for the life sciences sector, including freedom to operate, opinions, patent landscape analyses, patent and brand protection, licensing, development, and commercialization agreements, clinical trial protocols and agreements, patent litigation, risk management and product liability issues, pharmaceutical regulatory services, pricing and reimbursement issues, employment and labor law, competition and antitrust, and corporate and commercial services such as mergers and acquisitions, joint ventures, spin-outs, and initial public offerings. Whether working with startups, research organizations, or large pharmaceutical companies, the Gowling WLG team has a tremendous depth of life sciences experience. We are immersed in this sector and can guide companies through every stage of the product life cycle, as well as development of the company itself. Our expertise provided us with the opportunity to play a role in the response to COVID-19 by advising on a number of key projects relating to novel vaccines. These projects are the latest in a number of pieces of COVID-19 related advice provided by our life sciences team, demonstrating our extensive sector expertise with full service capabilities. To provide the comprehensive service essential to working in the life sciences industry, our leading patent prosecution, litigation, competition, and transaction and regulatory teams are here to provide strategic input to help life sciences companies maximize value, mitigate risk, and address any issues that arise. We are very much looking forward to this session today, and if you have any questions, please feel free to reach out to myself or any member of the Gowling WLG Life Sciences team. I will put my email in the chat. Let's begin our session, and our first company presenting is Bold Therapeutics with CEO Russell McAllister.
Thank you very much, Lisa, and thanks to Life Science BC for organizing this event. Can everyone see my presentation? I am Russell McAllister. I'm CEO and founder of Bold Therapeutics. We're an oncology company that's focusing initially on advanced GI cancers. We have a first-in-class small molecule therapeutic that significantly improves outcomes in difficult to treat cancer patients And I'll be sharing some new data today that we recently generated. Our compound works in combination with a wide range of different anti-cancer agents and in a broad range of different indications. We now have compelling phase 2 clinical efficacy data that suggests a breakthrough therapy designation and a single pivotal to approval by 2025. We have clean phase 1 and phase 2 safety data, orphan drug status in both gastric and pancreatic cancers, and as I mentioned, the potential for breakthrough therapy in colorectal cancer in the next couple months. Non-clinically, we've shown consistent and repeated efficacy across a wide range of models, and we're highly synergistic with other anti-cancer therapies. This is first in class, so you wouldn't have seen anything like it. It has a unique mechanism of action. As a result, minimal competition, and we have robust and recent global IP protection, in part courtesy of Gowlings. So thank you for your contribution. We're a relatively young company, but very fast moving. I think many of you have seen our story before, but we started in 2018, and we're now unveiling some phase 2 data that we'll be presenting at one of the cancer conferences in early 2023. We have one licensing deal already in South Korea, and we expect to strike another regional deal, likely Europe or Japan, in the next couple months. The design of our current study is a seamless adaptive phase 1b/2 study with 4 arms: colorectal, bile duct, pancreatic, and gastric, second line or later. And based on the early strength of the colorectal data, we added an additional colorectal third line or later arm, which I'll be talking about in just a minute. We're also adding a first-line esophageal cancer arm in combination with IO inhibitors based on some recent non-clinical data. Our trial, our phase 2 study is almost complete. We've enrolled colorectal, pancreatic, and bile duct cancer patients, and we're about halfway done with gastric cancer, which is ultra-orphan in Caucasians and North America and Europe, quite prevalent in the Pacific Rim. We presented our phase 1b data at ASCO this past June. Very encouraging data. Our drug, BOLD 100, is well tolerated in combination with FOLFOX, with no unexpected grade 3 or 4 treatment emergent adverse events. And it was safe and tolerable for an extended number of treatment cycles, with one patient up to 15 2-week treatment cycles. The early efficacy was exciting, but in a relatively small sample size, 19 patients, we showed a clinically meaningful improvement in progression-free survival versus the benchmark drugs. And a subgroup analysis in third line or later colorectal was particularly encouraging, as we were tripling the benchmark. We've also had some outlier responses in late-stage patients, which is particularly exciting because that's unexpected in that patient population. And much to my pleasure, this continued in phase 2. We continued to see excellent safety and tolerability, and the efficacy didn't worsen as I expected it would, but instead improved, going from the 1b into the 2. In addition to compelling data in colorectal that I'll be sharing in just a second, we also have encouraging data in both bile duct and gastric cancer. And perhaps just as importantly, we've seen numerous complete and partial responses in populations where those responses are quite rare, particularly in patients that previously failed on the other part of our drug combination, FOLFOX. So this is the data that we're very excited to share. The 2 approved drugs in the class, Bayer's Stivarga and Taiho and Servier's Lonsurf, are there on the right. This is the data off of which they were approved, with progression-free survival scores of roughly 2 months. And the data from our patients to date are with progression-free survival of 5.9 months, with strong statistics around that. So the remaining patients on study had the ability to move that only about 0.1. So the success for this study is now locked in. Not shown on this chart, but important, is our overall response rate, which is 500% better than these 2 drugs, and our disease control rate, which is twice as good. We're in the high 80s. These drugs are both in the mid 40s. This is a remarkable achievement because I never would have expected these results going into the study. We were looking for an improvement of about 30 or 40%. Obviously, this is a magnitude better than that. Mechanistically, this drug has a unique mechanism of action through GRP78, which is the master chaperone protein for the ER stress response, which is a common survival pathway across all cancers. This gives the molecule significant breadth of utility in the anti-cancer space. The left panel here is in treatment-naive pancreatic cancer patients and shows both monotherapy activity in blue as well as combination activity in green. And what you'll see here is that as a monotherapy, this drug is often better than best-in-class drugs, and in combination, it is always better. The right panel is multiple myeloma data, so a liquid tumor indication, and shows resistance breaking in combination with proteasome inhibitors. In multiple myeloma patients receiving the proteasome inhibitors inevitably develop resistance against these agents. The left panel here shows breadth of utility. There's a wide range of different cancer cell types and a number of different drugs, and we are synergistic or additive with almost all of them. The right panel shows that this is not just about resistant cancers, but in models of cancer where the cancer cell line is sensitive to the therapy, such as the N87 cisplatin sensitive line shown here, we significantly improve those responses. From an intellectual property standpoint, we have robust and recent IP. The US was territory of first prosecution. Those all issued some time ago, and our Japanese patents just issued. So we're excited about that, with patent expirations not until 2037 or 2038. From a manufacturing standpoint, we're almost at commercial scale with 2 to 2.5 kilogram lots and tech up to 10 kilogram lots. We do our drug substance in Europe and our drug product manufacturing in the US, although we're exploring moving that to Canada. From a capital strategy perspective, we've gotten this far on about $12 million, although it gets substantially more expensive from here with a potential pivotal phase 3 study starting next year at a cost of about $25 to $30 million. And we'd like to run a number of additional phase 2 studies in multiple myeloma and bladder cancer. We also have a pipeline of other interesting metallotherapeutics. Colorectal cancer is a significant problem that's untreated. It's a large market, and we think our drug is going to be very competitive in that space. We'll be presenting at ASCO GI in January, closing a second regional licensing deal imminently, as well as a large capital round, initiating additional studies, and moving towards an IPO in late 2023. And with that, I'd be happy to take any questions.
Thank you, Russell. Our second company presenting in this session is Mesintel Therapeutics, and I would like to welcome Jonathan Jafari, CBO of Mesintel.
Great, thank you, Lisa. I'm John Jafari. I'm the Chief Business Officer of Mesintel Therapeutics, and we are decoding fibroblast biology in order to create superior therapeutics. And I'd like to thank Lumira Ventures, LSBC, and Gowlings for sponsoring this session. So what are fibroblasts and why should you care? So fibroblasts are typically known as a connective tissue, a structural cell, which are found in basically all of the organs in your body. They were largely thought to be innocent bystanders in disease, but over time, we've discovered through 30 years of research in Michael Underhill's lab, my co-founder, that really these cells do have a prominent role in specific disorders, including sarcoma, where they are the cell of origin for these rare cancers, which I'll get into a bit later. The tumor microenvironment, the tumor actually recruits the fibroblasts to the tumor, where they shut off the immune system and make IO therapies inactive. A major portion of inflammatory disease, fibrosis, they have been very much implicated in the progression and the continuation of fibrotic disorders, and also in rare diseases. So there's been recent studies that have shown in disorders such as scleroderma that they are the major cause of the disorder. And we look to really have a deep understanding of the biology in order to develop novel therapeutics that shut off the programs which are causing the disease activity. So our target discovery platform comes from Michael Underhill's lab at UBC. It generated novel fibroblasts to understand what's going on with the fibroblasts, where they go, and whether they're active or quiescent. And we use single cell sequencing to identify the programs and activities of these cells and what they're doing. It allows us to understand the basic biology of the local environment, and we're looking for different ways to make them quiescent in order to improve the outcomes in various disorders. So our lead program is in fibrosis. This program is supported by the Stem Cell Network, Fueling Biotechnology Partnership Program. We've identified a number of novel therapeutic targets in order to potentially make these fibroblasts quiescent in the context of different fibrotic disorders. And we are also actually looking at the plasticity of these cells to actually make them potentially regenerative. This market's relatively large. There are 2 products that are approved, but they have a very limited efficacy and they have no survival benefit. And these patients typically die upon 3 to 5 years after diagnosis. We are currently looking at IPF, non-alcoholic steatohepatitis or NASH, and also rare fibrotic conditions such as systemic sclerosis and PSC. And so sarcoma is our second program, and these tumors largely affect adolescents and young adults, really impacting them at the sort of high points of their lives. And to date, there's been very limited efficacy in terms of the therapies that have been improved. They're mostly chemotherapies. And we feel that by really understanding the deep biology of these tumors, we'll be able to have successes, just like lung cancer, where you've got sort of 15 different subsets where there's much improved efficacy, as well as breast cancer. We are currently usually using genomic models which recapitulate the human disease, both in terms of the histology and the outcomes. And we have identified some novel targets, initially looking at some small molecule programs that we're testing in vivo. And we also have identified a novel surface marker, which we hope will be a potential bispecific program, and we'll be validating that target shortly as well. So in terms of the pipeline, I talked about the common fibrogenic targets, also immuno-oncology, sarcoma, followed by breast cancer. And then our proprietary discovery platform have identified a number of additional targets that impact the different fibroblasts and their function in various rare disorders that I mentioned, such as scleroderma, autoimmune disorders, such as IBD and Crohn's, ophthalmology, as well as aging. So as many of you probably are aware, as you age, there's a higher impact of various disorders, including fibrosis and tumors. We look to improve upon those outcomes. So our business model is based on the novel platform. We have a massive amount of data and analytical capability in terms of our proprietary platform. We bring in novel data from external sources, so human data that's publicly available, as well as different data sets that are available with different partners. We are developing our own programs currently, and we look to out-license the larger indications at specific inflection points. And we're also looking to validate the platform by pursuing platform partnerships with larger pharmaceutical partners, and we're in active discussions with a number of different companies. And we also use our platform partnerships to create different partnerships in terms of unique human biology data banks. So we are a pre-seed company, and we're asking for about $2 million to support the next 2 years of development, support our business development efforts, to identify partnerships, both on the out-licensing and the partnering of our platform, as well as in-licensing human data, formalizing our scientific and business advisory board, development and validation of our platform, complete IND-enabling studies for the sarcoma program, and also the preclinical studies for our priority indications. We are currently a member of the LSBC Investor Ready program, as well as CDL Advanced Therapies program. And we hope with all of the help that we're getting from a lot of different groups, that we will be able to raise these funds and move these programs into the clinic. The team is Dr. Michael Underhill has been studying these cells for about 30 years. Leslie Hill's the COO, and she's been doing a lot of the biology. Wilder Scott is the computational expert on the team. And myself, I'm more of a strategic business development person working in oncology and other rare disorders. And with that, I'd like to thank everybody today for attending the session. Again, thank you, Lumira. Thank you to LSBC and Gowlings, and I look forward to any questions that you may have. Thanks.
Thank you, Jonathan. Our next company presenting is Q Biologics with Dr. Hal Gunn, CEO.
Hal, I'm just going to jump on really quickly and let you know that your camera and microphone are off.
I'm sorry, thank you.
Not a problem.
There we go.
And if you wouldn't mind just going full screen.
Oh, thank you. As I had mentioned, Q is uniquely positioned. Our platform restores the full depth, breadth, and balance of immune function by restoring multiple cells and multiple pathways. It's designed not to force the immune system in one direction. It's designed to restore the full balance of immune function. Our platform is made from inactivated bacteria, and we expect a safety profile similar to vaccination, which is what we've seen in the 360 patients we've treated to date. Due to the unique nature of our platform and its anticipated safety, we can apply the platform not just to the treatment of disease, but to the prevention of disease, the maintenance of health, and the extension of healthy longevity. As you know, immune dysregulation underlies a wide range of diseases, from cancer to infection to chronic inflammatory diseases. And because of the nature of our product, we have the potential to treat and prevent all of these important disease categories, as evidenced by proof of principle studies in animals and for phase 2 clinical studies. By restoring health rather than just simply treating disease, we believe that our platform represents the future of medicine. We've raised $50 million US to date. We have compelling data from 4 phase 2 trials. We have a very good safety profile, as I mentioned, robust patent portfolio with 67 granted patents. We have a strong team, and we're really excited about delivering on the promise of our technology. We're seeking an additional $25 million of funding to finance multiple parallel randomized controlled trials. The simplest way of thinking of our technology is innate immune vaccines that target the restoration of innate immunity and all the cells and roles of innate immunity in a targeted diseased organ. And in doing that, restoring adaptive immunity as well. Our platform works through trained innate immunity, activation of myelopoiesis, and organ-specific chemokine release. It's self-administered by simple subcutaneous injection. Our platform is unique in a combination of 7 ways. As I mentioned, it's organ targeted. It works through multiple cells, I mentioned the safety. We've demonstrated synergy with all the cancer treatments we've tested it with, and it has application both in prevention and treatment across a wide important diseases. Here's some data from our study in Crohn's disease. This is RNA sequencing data looking at gene expression, and these are from biopsies of colonic tissue. Here we're looking at a range of different cytokine-mediated signaling pathways. You can see they're elevated at baseline, and they come back down to normal by week 16, and that's sustained through to week 26. You can see the p-value here, which is very low, and that's because we see this same pattern across dozens of different genes related to cytokine-mediated signaling. Cell migration is also important in inflammation, and you can see this same beautiful pattern across all of these genes related to cell migration. And again, as a result of that, a very low p-value. Neutrophils drive inflammation, and here are the genes related to neutrophil signaling. And you can see in the GI tissue, after treatment with our immunotherapy, we get a rebalancing and normalization of all of these genes. This is a study we did with Dr. Rebecca Auer, who's a leader in the field of post-op immune suppression. Surgery suppresses immune function, in fact, paralyzes natural killer cell function in the days and even weeks following surgery. That's, of course, very important in cancer because you're doing cancer surgery to try to cure the patient. But if the surgery paralyzes immune function, it can increase the risk of metastases. You can see both in lung cancer models and liver met models a substantial reduction in metastases. We received Health Canada approval and ethics approval for the initiation of this randomized placebo-controlled trial, 115 patients that will be initiating this month. The study will look at the potential of our therapy to relieve post-op immune suppression, clear circulating tumor DNA, and improve progression-free survival, which is the approval endpoint in this disease. There aren't any current treatments that work in this context, and we're very excited about moving forward with this study. We have a second recently received Health Canada and ethics approval, and that's a study looking at the potential relieve immunosenescence, which is the decline in immune function with aging. We are very excited about this study. It's a 72-patient study in people over the age of 65, looking at the potential of our treatment to restore normal immune function and to improve outcomes as we age. That decline in innate immunity increases the risk for cancer, infection. That study will also be initiating shortly. In blue, you can see the dashed line is our current funding. Those two studies that I just mentioned to the end of 2020. We'd like to raise $1 million to extend our run rate to the end of 2025 and to start a randomized controlled trial in inoperable pancreatic cancer. So we have a really great team, and we're really excited about moving our technology forward and a strong board. And so in summary, we have a very broadly applicable platform for both prevention and treatment. It's a very innovative approach, a very large opportunity across a range of important diseases, robust clinical data to date, strong IP that we thank Gowlings for achieving. And I look forward to connecting with anybody who'd like to learn more about Q Biologics.
Thank you, Hal. Our fourth company presenting in this session is Therapeutics, and presenting today is Dr. Amy Striltruck, co-founder, director, and CEO.
Thank you very much for that introduction. I'm just getting my screen up here. All right. It's such an honor to be speaking to you today alongside these other companies doing impressive and innovative work. So thank you for all who organized this session. My name is Amy, and like Lisa said, I am co-founder and CEO of a new venture called Therapeutics, which is harnessing nanomedicine technologies to create better therapies for coagulation disorders. 1 in 100 people has a bleeding disorder. 1 in 4 people dies from thrombosis. Disruptions in the balance of pathways that control blood clotting causes many prevalent and dangerous conditions. Disrupted coagulation is responsible for heart attacks, strokes, acute inflammation, and of course, bleeding disorders. Together, these conditions represent a huge market that can be addressed with RNA therapies, the benefits of which include high specificity and long duration of action. The long-lasting aspect of RNA therapies is especially relevant for people with bleeding disorders who need protection from bleeding for their entire lives. Within the category of bleeding disorders, there is a heterogeneous population that is underserved by current therapies. Most treatments on the market are limited to select patients, those with severe forms of hemophilia A, B, or VWD. And this leaves many unprotected, such as those with mild or moderate bleeding, a category that most women with bleeding disorders fall into, those at a higher risk of thrombosis, which again can often include women if they're taking oral contraceptives, and finally those with rare bleeding disorders or those for whom the cause of bleeding is unknown. Even for patients that are eligible, the current standard of care has limitations. Protein and small molecule-based therapies must be given every few days or even multiple times a day. Some treatments can overcompensate and cause patients to clot too readily, posing a risk of thrombosis. And in 15% of patients, the standard of care causes immune reactions that prevent future use of these or other therapies. These limitations prevent too many patients from accessing the protection that they need. Though the first thing that comes to mind with bleeding disorders may be the frightening prospect of a major medical emergency arising from everyday accidents, this is just one of the ways that people with bleeding disorders suffer. They also end up frequently hospitalized, potentially undergoing surgeries to mitigate the damage done by bleeding. Frequent bleeding episodes may lead to iron deficiency and anemia, and consequently a lower participation in school or work. And finally, chronic bleeding into joints leads to eventual disability, including the frequent visits to hospitals and outpatient clinics, prescriptions for pain medication and antidepressants, and the cost of treatment for bleeding. The annual healthcare cost for a patient with a bleeding disorder in the US is hundreds of thousands of dollars and rises as mobility is reduced. In order to address the burden of bleeding disorders, long-lasting therapies that provide stable protection from bleeding are needed. RNA therapies last for weeks instead of hours or days like current treatments. This can reduce the treatment burden and improve adherence while protecting against bleeding so that patients can engage fully with life. We are employing RNA technology to correct coagulation disorders by controlling the relative amounts of proteins in blood. The instructions delivered to cells by therapeutic cargoes like siRNAs typically last for 3 weeks after a single dose. Using siRNA also allows us to access new targets. Most bleeding disorders, though not all of them, are caused by a deficiency in the proteins that help form clots. For this reason, most treatments focus on replacing the missing protein or promoting clotting downstream. Unfortunately, these treatments are limited by the immunogenicity, short half-life, and thrombotic risks that I mentioned earlier. We are instead restoring the balance between clot formation and the separate process of clot clearance. By suppressing this pathway, we have shown that it is possible to reduce bleeding after injury back to normal levels and even prevent spontaneous bleeding episodes in gold standard models of bleeding disorders. Because we are focusing on the big picture balance of clot formation and clearance, this strategy will work in all bleeding disorders, regardless of the underlying clotting deficiency. Building on our preclinical results in gold standard models of bleeding disorders, we are advancing into IND-enabling studies with the finalized components that we expect to use in humans. This work is being supported through pre-seed funding and academic grants. As we look towards a fundraising round later in 2023, we will begin planning clinical trials focused on rare bleeding disorders, where there is an enormous gap between what patients need and the therapies available to them. We are also exploring the use of this therapy for heavy menstrual bleeding, a long overlooked problem that can significantly impact quality of life and affects a staggering 30 to 50% of women at some point in their lives. We also have patents filed on 5 other technologies, which we are in the process of licensing from UBC. The efficacy of these technologies in other types of coagulation disorders are being pursued by our academic partners and will be incorporated into our pipeline as we grow. Our nucleating team combines expertise in blood biochemistry and RNA therapy. We are joining an ecosystem of Vancouver-based companies that are developing complementary LNP technologies and working with partners to create the next generation of therapies for coagulation disorders. We're always looking for diverse perspectives and to expand our team, so if you'd like to learn more, please don't hesitate to reach out.
Thank you, Amy. Our final company for this session is Sustained Therapeutics with William Innate, President and CFO, presenting.
Thank you. Let's go here. Okay. Well, thank you, Lisa. I'm very excited to talk with you today about
Bill, you just exited your slide, if you wouldn't mind resharing it.
All right. Sorry about that. How's that?
Perfect.
Great. All right. Thank you. Again, very happy to be talking to you today about Sustained Therapeutics and our novel drug delivery system. I want to tell you 3 things today. So first off, the main focus, our initial focus, is in pain medications. Now, this is a huge market with high unmet need, particularly with non-opioid solutions like our product. Secondly, we have a unique platform technology which we're using to enable our pain medication, but there is potential for other indications, including in cancer and inflammatory diseases. And then third, in the pain space, we are a clinical stage company, completed a successful phase 1 trial. We have a phase 2 trial in postoperative pain underway, with 2 more phase 2 trials starting in the next few months. So talking about pain. Pain is a huge market, a very pervasive medical issue. Physicians have few treatment options at this point in time. What they are looking for is pain medication that is long-lasting, that's effective against all 3 types of pain, non-toxic, easily administered, and non-addictive. Unfortunately, there's not much out there now that does that. But as you can see from this table, our product meets all 5 of those criteria. We have a duration of 1 month from a single injection, 1 month of pain relief, as compared to hours or days with alternative products. Our product is good against all 3 kinds of pain: chronic, neuropraxic, and acute pain, has very low toxicity, and is non-addictive. And frankly, there's really not much out there except our product, which can make this claim. So tell you a little bit about the technology. This is an injectable drug. It is a viscous solution which is releases product over time. It basically is transforming multiple drugs potentially into novel locally injectable delivery systems. Now, here's how it works. There's two polymers that create this sort of viscous gel, which is injected into the tissue. An interesting thing is we can tune the release. The payload can be released over a matter of days, weeks, as with our pain medication, or potentially for months. And we're looking into indications in cancer with those longer-term tuning. Within the patent realm, we have got a freedom to operate analysis completed successfully. Our major IP has been patent issued in the United States and national phase applications in the rest of the world. And we have a second patent, which is US provisional and PCT. So a well-protected IP portfolio. Our pipeline, the pain medication, which we've developed, are the first three rows here. The bottom two rows are cancer drugs. In our pain medication, we've completed GMP manufacture at scale, completed a phase 1 product study in chronic scrotal pain, and have a post-operative pain trial in phase 2. And we will have further 2 phase 2 trials beginning in the near future. So talking a little bit more about this pain medication, low development risk, as I mentioned, it's been manufactured. We had great results in phase 1 and very promising results already in our phase 2 trial. The medication which we are using for pain is basically a reformulation of lidocaine. So lidocaine is generic. It's been around for a long time, very strong, a very low side effect profile. As a result, it's a 505(b)(2) pathway with the FDA. And what that means, of course, is that generally 505(b)(2) approval comes quickly and with a higher probability of success. It's a capital efficient clinical plan, fast endpoints, because the endpoints with pain are very, very clear. It either works or it doesn't. And so it's a short follow-up time, relatively small sample size. So that means that within the next 4 years or so, we should have completed our pivotal trials in pain, including phase 3 trials. This results in potential rapid value inflection points, IPO, out-licensing products, and so on. And in fact, we have multiple discussions underway with pharma partners at the moment. Quick look at our phase 1 results and efficacy. Small number of patients, but we did see efficacy. The takeaway here is we saw a clinically significant reduction in pain scores in 3/4 of all the patients in the study. And that is what you would expect because, of course, lidocaine is a well-known drug for pain relief. Being a commercial guy, I'm very excited about the commercial possibility. One of the pharmas that we have been working with has paid for an independent market research study, which was very well done. They found very strong commercial revenue drivers for the product. Talking to KOLs that they interviewed, very positive response. 80% respondents said this is a high met need. And also strong drivers for payers, positive healthcare economics, long-lasting benefit, and so on. In addition, this independent body projected the peak revenue for our first 2 indications, chronic scrotal pain, $300 million peak sales projected in the US, and for chronic pelvic pain, projected $1 billion. Management team, I'm very excited to be working with some tremendous co-founders. This is not our first rodeo. All of us have been in the industry for decades. We have, all of us have taken drugs from the bench through development, regulatory approval, and through to launch and on the market. Timeline, we raised a $3.5 million US seed round this year. We're going to be working on a Series A round in 2023. The use of proceeds for that round will be to complete our phase 3 trials. So again, very excited about this opportunity. We believe that we have low clinical and development risk, that we have low regulatory risk, fast time to market, low commercial or market risk, as well as very large markets with a lot of potential for follow-on line extensions and product extensions. And if you think about it, those are the key components of net present value. So what I'm saying is we are trying to maximize the net present value, maximize the value of our company. And we are confident that we will be able to value for investors, for physicians, for payers, and most importantly, for patients. So thank you very much.
Great, thank you, Bill. This concludes our presentations for this session, and I thank all of you for attending today. Wendy will now provide our closing remarks.
Thank you, Lisa. Firstly, thank you to Russell, Jonathan, Hal, Amy, and William for your great presentations. A really interesting mix of companies, and I look forward to following and continuing to be in touch and learning more about your developments as you go along. There's certainly a lot of very interesting innovation that you're all working on. I want to thank Gowling WLG for your support in bringing this session together, and specifically for your contributions, Lisa. You're a big champion of the life sciences sector in Vancouver, and we really appreciate it. Before closing off session 2, I would like to acknowledge and support the support that our event partners have brought that makes this event possible. So once again, our presenting sponsor, Lumira Ventures, our conference partner, Admare Bioinnovations, session sponsors, AstraZeneca, UBC, Genome BC, the Government of Canada, Trade Commissioner Service, Gowling WLG, Nimbus Synergies, and Novature. Our event sponsor, Innovative Medicines Canada, our networking reception sponsor, Gowling's, technology sponsor, Owen Wiggs, and event supporters, Berkeley, the British Columbian government, and Merck. Thank you all for your support. We really very much appreciate it, and it is what helps bring this event together. We all of our sponsors have exhibitor booths in the expo section of the virtual platform, so please be sure to visit them. And don't forget, there's an award for our most active networkers. Also, please remember to reach out to each other through the platform if you don't already know each other. There's lots of valuable connections that can be made at conferences such as this. We're now going to take a break from our featured presentations to allow you to stretch your legs, grab a bite to eat, and check out the expo. And I will see you back here at 12:45 Pacific Standard Time. So that's in about an hour, where we'll recommence our pitch presentations. Thanks very much, everyone.